Gibberellins are not known in American and British scientists until the 1950s. Rice farmers in Asia had long known of a disease that makes the rice plants grow tall but eliminates seed production. According to the World Scientific in 1998, a Japanese botanist named Kurosawa discovered a gibberellin called Bakini or foolish seedling disease of rice. Bakini disease is caused by the fungus Gibberella fujikuroi. In 1938, a Japanese botanist isolated two toxins in a crystalline form and named them gibberellin A and B, but because of the World War II and communication barrier the information did not reach the West. In 1955, England, Bryan et al. At the Imperial Chemical Laboratories independently obtained pure sample of a single gibberellin which was named as gibberellic acid. Later on, its structure was established by Cross et al. In 1961. It is now known that there are over 125 different gibberellins, which have been isolated and chemically characterized. However, not more than 15 different gibberellins have been detected from any single species of higher plant. The gibberellins are a large family of tetracyclic diterpene acids which have a common ant-gibberellin skeleton. The latter is itself derived from ant-green ring structure. Gibberellins are found in all parts of higher plant including shoots, roots, leaves, flower, petals, anthers and seeds. Gibberellins' activity has also been shown in plastids. In general, reproductive parts contain much higher concentrations of gibberellins than the vegetative parts. Immature seeds are especially rich in gibberellins and are most favored plants parts for the isolation of gibberellins and their study. In mature seeds, the gibberellins tend to form their derivatives. In plant, the gibberellins may occur in two different forms: free gibberellins and bound gibberellins.
Biosynthesis of gibberellins in plants. They biosynthesize in the apical tissues, and there are three main sites of their biosynthesis. First, in developing seeds and fruits. Second, in the young leaves of developing apical buds and elongating shoots. And lastly, in the apical regions of fruits. Gibberellins are tetracyclic diterpenoids made up of 4 isoprenoid units. Terpenoids are compounds made up of 5 carbon or isoprene building blocks, joined head to tail. The pathway of gibberellin biosynthesis can be divided into 3 stages, each of which is accomplished in a different cellular compartment. The first stage in gibberellin biosynthesis is the production of terpenoid precursors and encorine in plastids. The basic biological isoprene unit is the isopentyl diphosphate or the IPP. 2 IPPs used in gibberellin biosynthesis in green tissues were synthesized in plastids from glyceraldehyde 3-phosphate and pyruvate. Once synthesized, the IPP isoprene units are added successively to produce intermediates of 10 carbons, the geranyl diphosphate, 15 carbons, the farcinyl diphosphate, and the 20 carbons, the geranyl diphosphate, or the GGPP. GGPP is a precursor of any terpenoid compounds, including carotenoids and many essential oils. And it is only after GGPP that the pathway becomes specific for gibberellins. The cyclization reactions that convert GGPP to encorine represents the first step that is specific for the gibberellins. The 2 enzymes that catalyze the reactions are localized in the proplastids of meristematic shoot tissues, and they are not present in mature chloroplasts. Thus, leaves lose their ability to synthesize gibberellins from IPP once their chloroplast matures. The second stage in gibberellin biosynthesis is the oxidation reactions on the endoplastic reticulum from GA12 and GA53. In the second stage of gibberellin biosynthesis, a methyl group on encorine is oxidized to a carboxylic acid followed by contraction of the B ring from a 6 to a 5 carbon to give Ga12 aldehyde. Ga12 aldehyde is then oxidized to Ga12, the first gerabelin in the pathway in all plants, and thus the precursor of all other gerabelins. Many gerabelins in plants are also hydroxylated on carbon 3. The hydroxylation of carbon-13 occurs next, forming J53 from Ga12. All the enzymes involved are monooxygenases that utilizes cytochrome P450 in their reactions. These P450 monooxygenases are localized on the endoplasmic reticulum. Choline is transported from the plastid to the endoplasmic reticulum and is oxidized en route to chorinic acid by choline oxidase, which is associated with the plastid envelope. The third or the last step in gerabelin biosynthesis is the formation in the cytosol of all other gerabelins from Ga12 or Ga53. All subsequent steps in the pathway are carried out by a group of soluble dioxygenases in the cytosol. These enzymes require 2-oxoglutarate and molecular oxygens as co-substrates, and they use 2 molecules of iron and an ascorbate as cofactors. There are 2 basic chemical changes occur in most plants. First is the hydroxylation at carbon-13 on the ER or the endoplasmic reticulum or carbon-3 or both. 2 is successive oxidation at carbon-20. The principal physiological roles and effects of gerabelins in plants. Gerabelins are plant hormones that regulate growth and influence various developmental processes. It also stimulates stem elongation by simulating cell division elongation, as it overcomes dwarfism in mutants that have a mutation in the gerabelin synthesis pathway. It also breaks seed dormancy in plants, which require winter freezing. The endosperm of many seeds contain protein and carbohydrates, reserves upon which a developing embryo relies for energy and nutrition. These reserves must be mobilized and transported to the embryo. It also stimulates flowering or budding in response to lengthening days. It can induce seedless fruit development. It can delay senescence or the ripening in leaves and fruit. And also, it induces maleness or the sex expression in dioecious. They appear at low concentrations in vegetative tissues but at higher concentrations in germinating seeds. The transport of gerabelin in plants. It is made in the tissue in which it is used. Transport occurs through the xylem, phloem, or cell to cell. Phloem seems to be the most important transport route. Transport is not polar as it is for auxin.
Bioactive gibberellin can form local accumulation maxima, for example, in the root elongation zone and in the hypocotyl, which correlate with cellular growth. GA movement is necessary at multiple developmental stages, including germination, root elongation, the transition to flowering, and flower development. Long-distance movement of GA seems to be mostly restricted to its non-bioactive precursors rather than to bioactive forms. The first reports on GA mobility in higher plants appeared almost 50 years ago, establishing the presence of GA in the phloem sap and demonstrating the ability of GA to move through this medium. Since the early work was published, efforts to characterize and quantify GA movement in plants have been ongoing. GA moves inside the plant in both acro and basipetal directions, though it is more efficient in the former case. GA movement is clearly essential for several developmental processes. Also, evidences support the hypothesis that GA is a mobile plant hormone and that its movement is required for certain processes necessary for plant growth and development. One of the biological activities of GA is the induction of hypocotyledonous expansion following the flowering transition in Arabidopsis. Another example of an organ dependent on an external source of GAs are petals, which are dependent on the anthers as their GA source. A study carried out in Physarum sativum showed that it is possible to rescue the phenotype of GA biosynthetic enzyme mutants by grafting them into wild-type plants. Results pointed to GA20 as the major mobile form in Physarum sativum. It is also identified in another studies that GA12 is the major GA of Arabidopsis thaliana and transported over a long distance through the vasculature. J12 moved through the xylem in a root-to-shoot manner and then in the phloem in the shoot-to-root direction to regulate plant growth. In addition to long-distance movement, GCMS analysis revealed that the ovaries had relatively high levels of the GA precursors GA12, GA15, and GA9, and that the sepals and petals had relatively high levels of the bioactive GA4. Application of gibberellins have been shown to increase herbage growth throughout the growing season, although there is evidence that the response is less marked in the main growing season in summer. Gibberellins can be used most effectively to increase rates of dry matter production in spring by enhancing the rate of stem and leaf elongation, and in autumn by rapid mobilization of stored carbohydrate reserves and reallocation of nitrogen to shoots at the expense of roots. The GA signaling pathway is a major target for conventional breeding and genetic modification to alter plant stature. Similarly, it is possible that morphological and physiological traits in both horticultural and floricultural crops can be modified to increase productivity by manipulating GA metabolism and signaling pathways through molecular approaches.
When the seed starts to germinate, the roots and shoots starts to grow. But what is the precursor for this to happen? The seeds produce gibberellin, which is a hormone that provides energy to grow. This hormone also stimulates flowering, and it also works with auxin for elongation. Here is an overview of the video. In history, it was discovered by Kurosawa in 1998, and gibberellin was called backbone or foolish seedling. In 1938, Japanese botanists isolated two toxins in a crystalline form, namely gibberellin A and B. In 1955, pure sample of gibberellic acid was obtained. The structure was established in 1961. Today, there are 125 different gibberellin known, and gibberellin was discovered that it is a large family of tetracyclic diterpene acids. Gibberellin is found in all parts of plants, in shoots, roots, leaves, flowers, and petals, anthers, and seeds. There are two forms of gibberellin known, the free gibberellin and bound gibberellin. Biosynthesis occurs in three sites, namely developing seeds and fruits, young leaves of developing apical buds and elongating shoots, and apical regions of fruits. It occurs in three stages, namely in stage A, which produces precursor terpenoid precursor, stage B, in which oxidation reaction occur in endoplasmic reticulum from GA12 and GA53, and in stage C, or stage 3, which is the formation in the cytosol of all other gibberellin from GA12 and GA53. The role of gibberellin in plants are as follows. It is a hormone that regulates growth and influences various developmental processes. It stimulates elongation. It breaks seeds dormancy in plants.
It stimulates flowering and budding, and it delays ripening in leaves and fruits. For transportation, it happens in xylem, phloem, and cytosol. It moves in acro and basopetal direction, and for the application, it increases productivity and reduces nitrous oxide emissions in grazed grassland. Further, in application, it enhances growth by enhancing stem and leaf elongation, and in autumn, it rapidly mobilizes stored carbohydrate reserve. It also relocates nitrogen to shoots for fruits. I hope you learned much for our video.